Good afternoon, everyone. My name is Nicolas Hurtado Bautista, and I'm currently a postbachelor student at Charles R. Drew University of Medicine and Science. And today, I will be sharing with all of you the research that I have been working on, which is e-cigarette vaping increases DNA damage and reduces senescence in triple-negative breast cancer. Now, TNBC, or triple-negative breast cancer, tests negative for estrogen receptors, progesterone receptors, and has an excess amount of HER2 protein. The lack of these specific receptors does not allow for hormonal therapies to be effective, consequently making TNBC the most aggressive form of breast cancer. It is important to note that young African American and Latinx women are more prone to develop TNBC. Currently, electronic nicotine delivery system (ENDS) or vapes are very popular since it is believed to be healthier than smoking cigarettes. But our study focuses on the free radicals and harmful chemicals, such as nicotine, that increases DNA damage and reduces its cellular senescence response, leading to tumor proliferation. Senescence describes the process of a cell's deterioration with age, serving as a protection from tumors. So we hypothesized that the nicotine damages this tumor-suppressive response and increases the chances for young women to develop TNBC. For our experiment, there are three different groups, each containing a total of 5 nude female mice. The first group had a vape from e-cigarette with nicotine. The second group had vape from e-cigarette without nicotine. And the third group served as a control with no vape exposure. All the mice were exposed to vape within a vaping chamber for 12 hours, 3 times per minute, for 14 days. Post-exposure, mice were subcutaneously injected under their mammary pads with TNBC cell lines MDA-MB468 and HCC70. After a total of 90 days, the mice were euthanized and the tumors excised to compare the effects of e-cigarette on tumor growth. And an RNA sequence analysis was done on a xenograft to understand the differential gene expression patterns of the tumors. On the left-hand side, you have the tumors with the cell line MDA-MB468, and on the right are the tumors with HEC70. The first row, you have the tumors that did not have nicotine exposure, with the last two being the controls, and the bottom row, you have the tumors that did have nicotine exposure. From its appearance, you can see that the tumors with nicotine exposure are relatively larger than the ones without nicotine exposure. A further analysis was done where we took the volumes of these tumors, and we see that the tumors with nicotine exposure had a greater volume than the ones that did not. And if we look at our HCC70 tumors, we see again the same pattern where the tumors with nicotine exposure appear larger than the ones that did not have nicotine exposure. Another analysis was done where we took the weight of these tumors, and the tumors without nicotine exposure weigh less than the ones that did have nicotine exposure. So now we have a heat map where we have our three groups, the first one being our slow growing tumors, the middle group being the tumors just with e-cigarette vape exposure, and the last group being the ones that had nicotine exposure. On the right is just a list of genes, and where the ones circled are genes that are related to senescence, such as CAT5, SFA1, and UBN1. In the areas that are red, the red means that the specific gene is upregulated, or in other words, that there is high gene expression. Then in green, these areas are the ones that are downregulated, where there is minimum gene expression. So you can see it makes sense that in our slow-growing tumors, these senescence genes are upregulated, right? Because senescence is occurring here. It's protecting from tumor proliferation. Although once we get to our tumors with nicotine exposure, these genes are downregulated, meaning that senescence is not occurring here, so tumor proliferation is actually occurring. Again, we take a look here, we have our gene expression levels where we see the same pattern, where in our slow-growing tumors, there is higher gene expression than in our e-cigarette and nicotine exposure. And we have a statistical analysis where our p-value, right, shows that our data is significant. And in the graph right here, showing that again, our senescence genes are downregulated when there is nicotine exposure compared to our slow-growing tumors. It is also important to mention that TP53, which is a tumor-suppressive gene, is again downregulated, meaning that there is not much protection from these tumors when nicotine is involved. So in conclusion, we want to demonstrate a strong signature of reduced expression of senescence genes. From our data, we hypothesized that the e-cigarette actually increases DNA damage and reduces DNA repair and senescence in triple-negative breast cancer. Since these vaping products are relatively new, there are not much studies demonstrating the health effects from these vapes. And it's important that our findings bring awareness and that we further allow for the study of more health effects from the usage of these e-cigarettes in order to protect our communities. All in all, I want to thank the Urban Health Institute of Charles R. Drew, National Institute of Minority Health and Health Disparities for funding this research, and Xavier University of Louisiana for allowing me to present my research. And thank you all for listening. And if there are any questions, hear my evil. Once again, I am grateful for your time, and I hope all of you take care.